an email to the speaker. Now we present the next keynote presentation. The speaker is Professor Thomas Webster from Hebei University of Technology, United States. This is also another video presentation. I introduce briefly the biography of Thomas Webster. Thomas Webster degrees are in chemical engineering from University of Pittsburgh in 1995 and in biomedical engineering from RPI in 2000. He has served as a professor at Purdue, Brown, and Northwestern universities, and has formed over a dozen companies who have numerous FDA-approved medical products currently improving human health in 30,000 patients. His technology is also being used in commercial products to improve sustainability and renewable energy. He is currently helping those companies and serves as a professor at Brown University, Civetta University, Hebei University of Technology, and others. So we can start with the video presentation of Thomas Webster, entitled The Impact Nanomedicine, Orthopedic Nano Implants with No Failures and Still Company.
This is Professor Tom Webster, and thank you to the conference organizers for inviting me to give a talk. I have entitled today's talk, Human Nanomedicine. So, of course, I will emphasize magnetic materials, but I also wanted to emphasize something that a lot of people don't talk about, and that is human clinical trials with nanomaterials. Of course, we are used to talking about in vitro studies, in vivo studies with nanomaterials, but we don't emphasize too much human nanomedicine. So I've been very fortunate throughout my career to spin out a number of companies, some of the logos of which you can see on this slide, who are commercializing and have FDA approved nanomaterials that are in humans. So I want to emphasize that research during this talk while emphasizing the value of magnetic nanomaterials in medicine. So, of course, as I just mentioned, I spin out a lot of companies from my lab who have FDA approved implants that are in humans. So I need to give disclosures for these companies because I'll be talking about their work and I have a financial interest in those companies. So a great place to get us all thinking about nanomaterials, magnetic materials that can be found in humans is really to think about what is wrong with our healthcare system. What is it we are really trying to solve? And fortunately or unfortunately, there's a lot wrong with our global healthcare system. COVID-19 totally changed our lives. And really, this to me is a global healthcare failure. Our inability for healthcare systems around the world to deal with this virus highlighted so many problems in our healthcare system and highlighted why we need better materials from disease prevention to diagnosis to treatment. Clearly, if our only solution to a healthcare problem like COVID is to tell people to stay home, don't go to the office, don't go to the lab, don't go to a concert, don't travel, just stay home. If that's our solution, we have a failed healthcare system. Now, of course, many of us in the biomaterials area, we've been talking about the other failures on this slide before COVID existed. Overcrowded hospitals, barriers to even entering a hospital or doctor's office, medical devices that fail, and overdependency on drugs to treat all of our healthcare problems, treating every patient the same, having a reactionary healthcare system versus predictive healthcare system. And of course, we know there's so many inefficiencies in our healthcare system that drive up costs. And as we add more and more people on Earth who need healthcare with a growing global population, our costs will just skyrocket. And our whole healthcare system will be even more inefficient unless we come up with solutions. So 25 years ago, myself, many other people turned to nanomedicine for some answers. And included in nanomedicine are magnetic nanoparticles. So nanomedicine is a subdivision of nanotechnology where you're using small materials, particles, nanotubes, nanotextured surfaces to control cell response, to improve disease prevention, treatment, and diagnosis. So 25 years ago, we had the simple idea that cells would respond differently to nanomaterials compared to what we were implanting then, which we often call conventional materials or micron structured materials. We have since learned that not only do cells like bone cells, cardiovascular cells, soft tissue forming cells, neurons, they all respond differently to nanomaterials, but so do bacteria. And you can use nanomaterials alone without antibiotics to kill bacteria. You can also use nanomaterials to kill viruses. So I wanted to start with just a quick summary of how nanotechnology really helped us get through COVID-19, hopefully to be better prepared for the next pandemic, because there will be another virus pandemic that comes along. And I can't imagine us being less prepared than we were in 2019 when COVID struck the world. So you can think of nanotechnology used towards fighting COVID again in terms of prevention, in terms of diagnosis or therapy or treatment. In terms of prevention, nanoparticles are found in masks. Nanoparticles of metal oxides, when they get wet, they release reactive oxygen species. Those reactive oxygen species can break down the membrane of SARS-CoV-2 or any virus. So not only can you have a mask that traps a virus, but it can kill the virus. We also have materials that you can coat on everyday surfaces: aluminum, titanium, stainless steel. These are doorknobs, kitchen appliances, steering wheels of cars. You can put nanomaterials on those surfaces to keep bacteria from attaching, and if they happen to attach, to kill them, passivate them through the same mechanism. So we've been looking at things like selenium that you can put on a surface. Selenium can release reactive oxygen species and kill bacteria and viruses. We can also think of diagnosis. So you can use magnetic nanoparticles that are functionalized with regions to attach to SARS-CoV-2. You apply a magnetic field and you can now concentrate that virus. So you can find diagnostic kits that use magnetic nanoparticles, and they improve detection sensitivity and speed. The other area that I've been involved in all of these areas, but another one is therapy. Can you develop a nanomaterial that can wrap around the virus, passivate it, keep it from replicating? Here's another way we've been trying to help with COVID detection, and this is through a company called Quarkson, where we have developed a breathalyzer. So if you think you have COVID or you've been exposed to COVID, you can breathe into this breathalyzer, as shown here, and our sensor can tell you if you have SARS-CoV-2 in your breath. This can also work for other viruses like HIV, influenza, rhinovirus, which is the common cold, and other biomolecules such as those for Alzheimer's disease or bone diseases. So this is just an example of how we've created this sensor breathalyzer for detecting COVID. And how it works is through molecular imprinting. Take the virus, SARS-CoV-2, make an imprint onto polypyrrole that copies the geometry of the virus, therefore making it more able to attach to the virus. And once that attachment occurs, you have a change of conductivity because you have graphene, you have other semiconductor elements that basically change resistance. So I'm happy to show you some of our first clinical data where a COVID positive patient took our breathalyzer and it told them that, yes, they do have COVID. So they were able to determine that they had COVID from a traditional nasal swab assay. And here you can see how that resistance, conductivity of their once they breathed into a breathalyzer changed. So here's resistance on the y-axis down here, time that they were breathing into a breathalyzer. The green line is for the COVID positive patient. We compared it to other types of molecules to show that it was different. But here you can see pre-exposure. Then as soon as the patient took a breath into the breathalyzer, the resistance went down. The SARS-CoV-2 was captured in our sensor and the conductivity went up. So that enabled us to give them a positive indication where the other viruses did not, to say that you have COVID. So again, we're taking this idea, looking at a wide range of viruses. We're also looking at room detectors, putting these sensors in airplanes, in trains, to basically detect if you have COVID or SARS-CoV-2 in the atmosphere. Another project that we've been involved in is therapy. So if you test positive, you have symptoms, what are you going to do? And let me show you a quick video to show you how our nanomaterial works.
The self-assembled nanomolecule can attach to the virus surface protein. This would keep the virus from attaching to a cell, entering the cell, and replicating.
So clearly you can see the idea is very simple. We are developing a nanomaterial. The virus, SARS-CoV-2, all viruses are nanometer. So if you want to stop their replication, you have to be at the nanometer size range. We developed a molecule that can attach to SARS-CoV-2 and it blocks it from entering the virus, and therefore it blocks it from replicating. So what does our molecule look like? We call it the blanket. Here is the chemical structure. Essentially, it's two DNA base pairs, guanine and cytosine. We functionalize them together. When they get into an aqueous environment, they form all of these hydrogen bonds, form a ring-like structure around the virus, and we can have side groups, functional amino acids, that allow this molecule to then attach to the virus. What's shown here is lysine. Lysine is great for promoting tissue growth. We have a history of also using this molecule with a lysine side group to increase bone growth, increase cartilage growth. But if we replace that functional group with something that attaches to SARS-CoV-2, we can passivate SARS-CoV-2. And we've demonstrated this in animal models, a transgenic mouse model, where we looked at how much SARS-CoV-2 was in the lung after 7 days. The y-axis is the amount of RNA, SARS-CoV-2 RNA. Of course, the control had a high level after 7 days of in the lung. We use our molecule as a nasal spray. We compared it to Paxlovid. Paxlovid is an FDA-approved therapeutic molecule. It reduces your symptoms if you test positive for COVID. However, a lot of studies have shown once you stop taking it, your symptoms return and your viral load returns. And this is because Paxlovid works inside a cell. Our molecule works outside the cell. As soon as the virus gets inside the cell, it's already started replicating. So we want to keep that from happening and passivate it, keep it from getting inside the cell. Nonetheless, we see in our doses that we were able to get mice to survive. Mice died in these other conditions, and they were statistically similar to Paxlovid. So different mechanism of action. We are hopeful that our molecule does not promote the return of viral load like Paxlovid does, and acts in a similar way to reduce viral load to get that patient out of COVID symptoms. So we're in the final stages of FDA approval for this molecule, and hopefully you'll be able to have an over-the-counter nasal spray to protect you or treat you if you detect positive for COVID. So now into a little more of human studies with nanomaterials. I showed you the sensor breathalyzer for COVID, but there's other human studies that I've been involved with that I want to share. And it has to do with not self-assembled chemistries like what I just mentioned, but nano-textured surfaces. Nano-textured surfaces we have seen, and other groups have seen this, can control cell responses without using drugs, can reduce bacteria colonization without using antibiotics. So you don't have to be concerned about generating antibiotic-resistant bacteria, and can also reduce inflammation, harmful inflammation that can occur around implants. So we've taken this idea and we've nanotextured spinal cages. We've nanotextured amputee devices, which I'll talk about in a little more in a second. We've nanotextured screws that are used to attach soft tissue like ligaments, tendons, muscle to bone. We've nanotextured vascular implants, and we've nanotextured catheters. So in all of these cases, if you create the right nanotexture, you can promote tissue growth, reduce bacteria colonization, and reduce inflammation. Now, I could spend a lot of time telling you why, and the community, not just my group and my companies, but the community has really assembled to try to understand why these large cells care about a nanotexture. And we could give you reasons where people have looked at nature and they've seen that insect wings have nanotextures, and these nanotextures reduce bacteria colonization. So obviously, through evolution, insects have figured out to have nanotextures to improve their health. So we're simply using these ideas on our medical devices to improve their performance. We also could point to biophysical models, things like finite element analysis, where research groups have looked at how bacteria membranes are too stiff to attach to all the contours of a nanotextured surface. Now, bacteria are about 1 micron in diameter. Mammalian cells are about 50 microns in diameter. Mammalian cells, like bone-forming cells, have a much more fluid cell membrane. It's bigger and it's more flexible. So they can, and they prefer to attach to these nanotextures. In fact, those nanotextures provide a three-dimensional environment, which is helpful for bone cells to grow. I could also point you to some of our own research where nanotextured surfaces interact differently with proteins that control cell attachment. As soon as you put an implant in the body, proteins absorb. And it's those proteins that determine whether a bone cell will attach and grow, or whether you can repel a bacteria or repel an inflammatory cell. So this very quick study using atomic force microscopy shows you that creating nanotextures on PLGA, a very popular polymer, putting fibronectin, a protein known to improve tissue-forming cell function. When you put that, allow it to absorb to a nanotexture, it absorbs in a fibrillar, more bioactive manner. When you allow that protein to attach to a micron textured surface, it absorbs like a hemisphere, a blob, a non-bioactive form. So you can use nanotextures to control protein interactions, both get more of that protein to absorb and make it more bioactive. In fact, we've created an equation where we can predict the size of a nanofeature you should put on an implant to improve its performance. We call this nano-optimized, and we've trademarked this name to basically work with people, work with researchers as well as companies that have implants that they want to reduce infection, or they want to reduce inflammation, or they want to improve bone growth. We can take your implant, tell you what the size of that nanofeature should be, and then help you create that nano texture. So that nano optimized trademark is something we're pretty proud of. It's been tested on over 24 implants, and it seems to work for polymers, metals, ceramics, and composites. So let me give you 3 quick examples of projects I've been a part of. They've spun out of my lab into companies. Those companies receive all the credit. They got FDA approval, and they are now in humans improving their function, as you will soon see. So the first company is called Nanovi. Nanovi takes orthopedic spinal screws and anodizes them. That anodization process is shown here. You put titanium into this anodization chamber, apply a voltage in an electrolyte, and you can create nanotextures. Tubular nanotextures, pores of nanotextures on these screws. You can change the dimension of those pores, the depth of those pores. You can do a whole bunch of things. But our very first animal study, we put this into an amputated limb rat model. So we worked with the FDA on getting FDA approval for this type of an implant. Here you can see the rat study, the X-ray, where the limb was resected, a screw was inserted. Some of those screws were anodized to have nanotextures, some were not. We immediately saw success of the anodized nanotextured implant, where after 3 days of surgery, the rat was putting weight on that implant, indicating that enough bone grew around it for the rat to put weight on it without feeling significant pain or discomfort. For the non-anodized implants, the ones without nanotextures, the rat was not walking on that implant upwards of 28 days. That was the length of our study. We took a closer look at this exit wound site to learn more. And what we saw also supported the use of nanotexturing in orthopedic screws. Here you can see with the screw that is primarily used today, and definitely was used back then. It basically has an open wound. So the screw that's coming out of the remaining limb after 14, 21, and 28 days, that wound is open. This is a prime spot for bacteria to colonize that wound, infect that wound. We routinely saw an infection. We saw pus formation. When we nanotextured that screw, the skin grew right up to the screw. It closed the wound, kept bacteria out, stabilized the implant, and essentially promoted bone growth. So NanoV now has 14,000 humans that have this implant. They are not using it as an amputee device, but as a pedicle screw. So here's the spine, a human spine. Basically, the pedicle screws are inserted, needs to stabilize the spine. Of course, you don't want infection. You don't want bone non-unions. You want bone to grow right next to this implant to stabilize it. So impressively, out of the 14,000 humans that have these implants, there have been no cases of failure. And that's much better than the industry standard, which is about 5 to 10% orthopedic implant failure, either due to infection, bone non-unions, loosening, or other complications. The second example, I collaborated with a company that's now called Syntex. They commercialize silicon nitride also as a spinal implant. Silicon nitride smooth right here is what they commercialized at the time. We helped them develop a process to create nanotextures following our equation of the right size of nanotextures. We then compared these in an animal study to titanium and PEEK, polyether ether ketone, which is the #1 implanted polymer used in the spine. Now, when working with the FDA, we wanted to see, are these materials, the nano rough silicon nitride, antibacterial, or do they reduce bacteria function? So they recommended to us, put bacteria on these implants, then insert them into the rat calvaria or the skull of rats. So we did that. After 3 months, bacteria inoculation, again, we put bacteria on these implants, we forced an infection, then we took histology stains. Here's the 4 materials. The purple is bone growth, the yellow is bacteria. Here is the surgical site, the circle that you can see. And clearly for titanium, we still see yellow. For PEEK, we see a lot of yellow. But when we change chemistries to silicon nitride, we don't see any yellow or very little yellow. So changing chemistry to silicon nitride reduced bacteria colonization. And in fact, the nano rough silicon nitride completely inhibited bacteria presence. There were no bacteria found surrounding this implant after 3 months. And of course, that left a lot of room for bone to grow. So we saw about 41% of that implant surface covered with bone, whereas the smooth silicon nitride was only about 15%. And as I mentioned, Medica is marketing this, selling this, using this as a spinal implant. Over 4,000 have been implanted in humans. Again, no cases of failure. The last example I'll give you is with a company called Tiber Medical. Tiber Medical has been using electrophoretic deposition that we developed, where you take titanium and coat it with ceramic nanoparticles. So we took titanium screws and coated them with nanometer large, so about 170 nanometer diameter calcium phosphate particles. Then also in B is 150 diameter, so getting smaller still, 130 nanometer particles. And then the smallest we tested was 110 nanometer particles. So we are coating this with smaller dimension particles to increase nanotexture. The smaller the particles, the higher the nano texture. We compared that to a plain titanium that was coated with plasma spray, calcium phosphate, and then just plain titanium. We saw incredible antibacterial properties. When doing so, we were able to decrease Staphylococcus aureus on the highest nanotexture surface. So the smallest diameter particle, highest nanotexture. That was significantly better than the industry standard of plasma spray. Not only for Staph aureus did we see that, but also Pseudomonas aeruginosa. We were able to decrease bacteria colonization, and we were able to do this for a drug-resistant E. coli, ampicillin-resistant E. coli, where we significantly reduced bacteria colonization. So gram-positive, gram-negative, and antibiotic-resistant bacteria can all be reduced when using these nanotextures. So over 15,000 of these have been implanted into the feet of patients with 0 cases of implant failure. So 3 different chemistries: titanium, silicon nitride, calcium phosphate. The similar property is they're all nanotextured, and they all have been showing 0 cases of infection. So wouldn't it be great if this is our solution to implant infections? So now let me spend some time talking about magnetic nanomaterials. So some of the materials I've talked about before are not magnetic. So let me give you an idea of how magnetic materials can give you the same proactive properties if you make them into nano compared to conventional or micron. And let me start with a cardiovascular tissue application. We're all familiar with heart attacks or myocardial infarctions. It's clearly a big problem in the world today. Cardiovascular disease is the #1 killer of people around the world, but unfortunately, there aren't many solutions. You can receive a heart transplant if your heart attack was too severe, but there's limited donors and there's limited transplants to even give you. You can think about cell-based therapies, which we do. Can you use stem cells from the patient and a material and regenerate a beating heart that then could be used for implantation? So you could think about bioengineered scaffolds, a cardiac patch, if it's a minor heart attack where you just use a patch to regenerate healthy heart tissue, or an injectable material, or a complete new heart. So here's an example that we have created: magnetic nanomaterials, iron oxide nanomaterials. We cultured what are called synoviocytes. These are beating cells of the heart, and we've been able to create a significant faster beating heart when using magnetic nanomaterials. So here you can see the heart beating, synchronous. We can match this with the natural heart beating, and it only is achievable if you use magnetic nanomaterials. If we use magnetic micron materials or non-magnetic materials, we're not able to achieve this wonderful beating heart, which could eventually, hopefully, be used in a human as a heart replacement or a cardiac patch to just heal a small part of damaged heart. In the brain, neurological magnetic materials. We've been very interested in looking at neural probes, materials that you can use to jumpstart damaged parts of the brain. You might have stroke, and if you have stroke, blood gets cut off to your brain, part of your neural tissue dies. So how can we regenerate healthy brain function where part of your brain died due to stroke? We do know that neural probes can be used. These can be inserted into your brain, apply a voltage, jumpstart those cells. But the big problem is these neural probes don't last that long in the brain. They get scar tissue to form around them. And that's what this image is showing. Here's the probe in the middle. where you have all this white scar tissue. Scar tissue is not conductive. So you need to reduce that scar tissue. So we've been looking at magnetic carbon nanotubes as a way to promote neural function and decrease scar tissue. In the interest of time, I don't have time to show you our in vitro data where we've looked at astrocytes. Astrocytes are cells that contribute to scar tissue. We've seen you can reduce their function using magnetic carbon nanomaterials. You can also increase the functions of neurons, obviously the cells that conduct electricity in your brain, using these magnetic materials, carbon nanomaterials. But the real study I wanted to show you was using carbon nanotubes, magnetic carbon nanotubes with stem cells. So we conducted a study with Professor Lee from Yonsei University where we established stroke. This is middle cerebral arterial occlusion for 1 hour. We then allowed the blood to reperfuse in these rats. And then at 9 hours, we injected our magnetic materials with stem cells. And then 1 week, 2 weeks, 3 weeks, 5 weeks, 8 weeks later, we measured motor function. We measured what happened to these rats. And I should tell you the reason for this study was that Professor Lee, who is an expert in stem cell technologies, was inserting stem cells alone into stroke-induced damaged brains. But those stem cells did not stay where the damage was. Those cells migrated to healthy cells, which makes perfect sense because healthy cells are producing cytokines, chemicals, to recruit stem cells. So she wanted an anchor, something that could keep the stem cells in place. And we knew from our work that stem cells attach to these magnetic carbon nanofibers, so we could use them to keep the stem cells in place where that damage is. And that's indeed what happened. So we have two different kinds of magnetic carbon fibers, hydrophobic, hydrophilic, but here's a section of the brain. The carbon fibers are black, nanofibers are black. They have the stem cells attached to them. And we saw that for the duration of the study, the carbon nanotubes did not migrate around the brain, but they stayed where the stroke was. And we could see where the stroke was using other kinds of imaging tools. So, of course, we looked at how those stem cells differentiated. And to make a long story short, what we saw was that the stem cells differentiated into neurons and not astrocytes or scar tissue forming cells. So again, here's the carbon fibers, the magnetic nanofibers. You can see the stem cells around them. We use different molecular markers to show they, in fact, transformed, differentiated into neurons, which is what we wanted in the area where we injected them. Functional recovery told us the whole story of the success of this approach. A treadmill, of course, if a rat has stroke, following this method, they're not able to take any steps on a treadmill, but using stem cells with our carbon nanofibers, the animal took more steps. Same thing with a reflex called the Vibrise limb placement. This also enabled the combination therapy to excel. So we used stem cells with carbon nanofibers, and the reflex function was enhanced compared to stem cells alone and stroke alone. And the last motor function I'll show you is the rotor rod, where again, the rat was able to stay on a spinning rod longer when we injected stem cells with carbon nanotubes, nanofibers, and not the stem cells alone or the stroke alone. So in the brain, there's also promise for magnetic nanomaterials. But I must say, having spent my career looking at carbon nanotubes in medicine, we really, as a community, need to be careful. Many studies exist out there which supposedly report toxicity of carbon nanotubes, but I beg you to take a closer look at these studies because the titles do not match the results. So we can't be alarmists, but we have to accurately report our studies because agencies like the FDA or other regulatory agencies will just look at these titles and think that carbon nanotubes are toxic. So let me give you two examples of where titles can lead you astray in terms of actual results. So this title, Carbon Nanotubes Introduced into the Abdominal Cavity of Mice, Show Asbestos-like Pathogenicity. So asbestos has often been compared to nanomaterials. Asbestos was used as insulation many years ago, decades ago. They are not nanomaterials, they are silica that have micron dimensions. And you got lung cancer, or people got lung cancer from installing this insulation because they were breathing those asbestos particles into their lungs. So this study basically injected carbon nanotubes into the stomach and then tried to see if asbestos-like cancer would result. Nobody was injecting asbestos into the stomach. So the route of entry is completely different and makes this title inappropriate and an alarming title and something that should not have done. And this appeared in Nature Letters. Another study that is inappropriately titled, Induction of Mesothelioma. Again, mesothelioma is the asbestos-like cancer by single intrascrotal administration of multi-wall carbon nanotubes in rats. Again, nobody who was looking at mesothelioma lung cancer was injecting asbestos into the scrotum of people. This administration route is completely crazy, and it should not be related to how people got mesothelioma. So again, please do your studies carefully, come up with titles that accurately represent your results. And the other thing that I notice in the literature all the time about carbon nanotubes, whether they're magnetic or not, is you have to carefully characterize your nanomaterials. As you know, sometimes catalysts are used to grow nanomaterials. Those catalysts can be toxic, and if they're left in the nanomaterial, it should not be surprising that your nanomaterial is going to be toxic. So you need to accurately characterize your materials and report that to the scientific community so that we can understand what the problem is. So let me leave you with one last example of how we are going to be more proactive in medicine using magnetic nanomaterials. And magnetic materials, I believe, can help us create more proactive materials in medicine. We can't have inert or reactive materials and methods in medicine. We can't always be reacting to your problems. We have to predict them, and we have to have more intelligent materials. And a lot of this gets to this idea of healthcare at home. As we learned during COVID, and we knew this was a problem before, our hospitals are overcrowded. The idea of a hospital is very old-fashioned, 17th, 18th century type idea that still exists today. We have so many technologies that have advanced, but yet implants have not, and healthcare has not. We should be able to determine your health wherever you are and solve your health problems wherever you are, not asking you to come into a hospital. So I'm going to skip by the sensor component here and just talk about something called 4D printing. We all are aware of 3D printing. You have a CAD program, put it into a 3D printer, it prints a specific device. We can build houses out of 3D printing, computers, furniture, you name it. We are using 3D printing. Same thing in medical devices. You can 4D print medical devices and make it 4D by adding the property of changing shape with time. So that fourth dimension in 4D printing is changing shape with time, wirelessly and on demand. So here's an example of a study from my research group. You can go to a YouTube video to see more about it, should you like. We are using magnetic graphene nanoparticles. You put that into your 3D printed material, whether you've 3D printed a flower, a hand, a circuit, a heart, and you apply near infrared. Near infrared will cause those graphene nanoparticles to heat up and change shape. So for a flower, you can make petals open up. You can make certain fingers appear. For circuits, you can block certain circuit pathways to enable switch on, switch off. Or you can squash your heart. So these graphene nanoparticles enable this to occur because they heat up under near infrared. So we're taking this idea in so many places. And let me show you a video here of a material that we made with graphene oxide nanoparticles. We've exposed that material to near infrared, and it starts opening up on demand whenever we want it to. We're taking this idea and we're treating scoliosis, curvature of the spine. If you know anybody who's had scoliosis, you know it's a painful surgery. Spinal rods have to be inserted many different times into the patient, and slowly the spine straightens. Obviously, with a material like this, we can use one surgery and have a slow straightening of the spine using this one material that's connected to the spine. Intervertebral disc, you can take this material, insert it into the disc space, cause it to swell. We know bone grows under increased pressure. So you cause it to swell, and that promotes bone growth. Other applications, stem cell delivery. Instead of lifting cells off of a petri dish, use the same sample you're culturing cells on, cause it to curl up, and now you can deliver it. So you don't have to lift those stem cells, which ends up killing some of them. The last one, sphincters. You can have sphincters that open and close. As you age, some of these sphincters get weak, and you end up with acid reflux, acid going from the stomach to the esophagus. So we're developing 4D printed sphincter materials that can keep the acid in the stomach and, of course, open up to allow food to come down. So many applications. We're at the very beginning of using 4D printing, but I hope in this presentation, I gave you an idea of how nanotechnology is being used throughout all of healthcare, some real human data showing success. And I hope I also highlighted magnetic nanomaterials, how they're used in the heart, can be used in the heart, can be used in the brain, and can be used in 4D printing, like these graphene oxide nanoparticles to on-demand change implant shape after it's been inserted in the body. So again, I thank all my startup companies, the funding agencies. I have a new book out if you're interested in reading it, but I really wanted to thank my students through the years of developing such wonderful research that now I'm trying to commercialize in the last part of my career. And please email me with questions. And again, I thank you for your attention. Thank you very much.